Good day, dear students. In this pre-recorded lecture, The thermal cycler, also known as thermocycler, is a laboratory apparatus most commonly used to amplify segments of DNA via the polymerase chain reaction or PCR. The device has a thermal block with holes where tubes holding the reaction mixtures can be inserted. The cycler then raises and lowers the temperature of the block in discrete, pre-programmed steps. The first PCRs were performed using multiple water baths or heat blocks that are set at the required temperatures for each of the steps of the PCR cycle. The tubes were manually moved from one temperature to another. And in addition to this hassle, before the discovery of thermostable enzymes such as the Taq polymerase, new enzyme had to be added after each denaturation step, further slowing the procedure and increasing the chance of error and contamination. So in this slide, you can see a picture of the PCR setup that scientists used to have before thermal cyclers were invented. Therefore, engineers started to look into inventing an all-in-one instrument, which is the thermal cycler, that would help automate the PCR process. In 1987, the first commercial thermal cycler, which is the TC1 DNA thermal cycler from Perkin-Elmer Cetis, became available with the ability to program heating and cooling of the samples using a metal block. In 1988, the first use of a thermostable enzyme, which is the Taq DNA polymerase in PCR, using the TC1 thermal cycler was reported. This paved the way for applications of PCR in a wide range of scientific fields, as well as innovations in thermal cyclers that revolutionized molecular biology researches. Since the introduction of the TC1 thermal cycler, significant technical progress has been made on various features of the equipment to improve PCR experiments. These thermal cycler improvements have resulted to better sample handling, more precise PCR optimization, faster PCR protocols, and easier PCR setup and enhanced accessibility. In the early development of thermal cyclers, the cooling system relied on a bulky plumbing compressor, which made it impossible to have a small footprint instrument. In the present day, solid-state Peltier blocks are utilized in thermal cyclers to both heat and cool by controlling the direction of an electrical current. Advanced Peltier systems can heat and cool the block at a fast rate, say for example 6 degrees Celsius per second, enabling fast PCR to complete more PCR runs in a day. Similarly, a heated lid is now a common feature of thermal cyclers to prevent evaporation and condensation of PCR samples during runs. Prior to the introduction of the heated lid, samples were overlaid with mineral oil to achieve the same purpose. In addition to being inconvenient and messy, oil overlays limited the amount of a sample that could be used in downstream applications because part of the sample had to be left behind to prevent carryover of the oil. The next important development of thermal cyclers at present is that they enable a more precise optimization of the PCR protocol, specifically on the annealing temperature. Since annealing of primers to the target sequence is critical to obtaining successful PCR results, the temperature for the annealing step often requires optimization. To examine different temperatures simultaneously, gradient thermal blocks were developed, which are designed to enable setting desired low and high temperatures around the theoretical annealing point at the two ends of a single metal block. Better than gradient technology is also now available, in which insulated separate metal blocks replace a single block, which allows more precise temperature control for faster optimization. Then, some thermal cyclers at present were developed to produce faster PCR protocols. Fast PCR refers to protocols that dramatically speed up overall PCR run times, typically reducing run times from approximately 2 hours to less than 40 minutes, thus saving time and increasing throughput. Thermal cyclers today are designed for easy programming of PCR protocols. PCR protocols often vary based on the DNA targets, the primer sequences, DNA polymerases used, and experimental goals. Therefore, thermal cyclers that are equipped with intuitive user interfaces, such as touch screens and easy programming features, enable faster and more efficient protocol setup. Recent advances also allow convenient access to thermal cyclers anytime and from anywhere using a mobile device or desktop computer. Connectivity to the cloud offers enhanced accessibility at your fingertips and freedom to create and share protocols as well as to schedule, to start or to stop, and to monitor PCR runs. Now let me move on to our next topic of interest, which is controls for PCR. As with any diagnostic assay, running the correct controls during every PCR run is essential for ensuring and maintaining the accuracy of the assay. In PCR, there are different types of controls that are used. These are the positive control, contamination control, negative template control, and internal amplification control. So we have the positive control, which contains the known target sequence to be amplified. Positive controls ensure that the enzyme is active, the buffer is optimal, the primers are priming the right sequences, and the thermal cycler is cycling appropriately. A negative control without the DNA, or also called a contamination control or reagent blank, ensures that the reaction mix is not contaminated with template DNA or any amplified products from a previous run. Meanwhile, a negative control with DNA that lacks the target sequence or the negative template control ensures that the primers are not annealing to the non-target sequences of the DNA molecule. Then, there is the internal amplification control. In some applications of PCR, an internal amplification control is included that contains a second set of primers and an unrelated target added to the reaction mix. These controls demonstrate that the reaction is working even if the test sample is not amplified. Amplification controls are amplified preferably in the same tube with a test reaction, although it is acceptable to perform the amplification control in a duplicate reaction mix if the control cannot easily be distinguished from the target product in the same reaction. This type of control is most important when PCR results are reported as positive or negative, negative meaning that the target sequences are not present. The amplification control is critical to distinguish between a true negative for the sample and an amplification failure or a false negative. Finally, I will be discussing about the PCR modifications. PCR has been adapted for various applications, several of which are used in the medical laboratory. Among the large and increasing number of PCR modifications are the following methods that might be encountered in the clinical molecular laboratory. These methods are capable of detecting multiple targets in a single run, using RNA templates, or such amplified products as templates, and quantifying starting template materials. In multiplex PCR, 2 or more primer sets designed for amplification of different targets are included in the same PCR reaction. Using this technique, more than one target sequence in a clinical specimen can be amplified in a single tube. As an extension to the practical use of PCR, this technique can save time and effort. The primers used in multiplex reactions must be selected carefully to have similar annealing temperatures and must be not complementary to each other. In this example, multiplex PCR is used to detect for the presence of 4 different target strains in one PCR tube reaction. This can be done by adding the reverse and forward primer set for each target strain into the reaction mixture. Each strain is detected by fluorescent signals released from the fluorescent dyes that are attached to each primer set. The next modification of PCR is reverse transcriptase PCR or RT-PCR. This was developed to amplify RNA targets. In this process, complementary DNA or cDNA is first produced from RNA targets by reverse transcription, and then the cDNA is amplified by PCR. The reverse transcription is accomplished through the action of the enzyme reverse transcriptase. Next is real-time PCR or quantitative PCR. qPCR is the technology for the quantification of nucleic acids. This method is a way of quantifying the amplification of a DNA as it occurs, which means it detects the accumulation of amplicon after each thermal cycle in real time. Real time implies that data collection and analysis occur as the reaction proceeds. The initial concentration of target sequence can be determined by a comparison with the amplification kinetics between the target and an internal control with known copy number. Next is nested PCR. Nested PCR was developed to increase both the sensitivity and specificity of PCR. This technique uses 2 pairs of amplification primers and 2 rounds of PCR. Typically, 1 primer pair is used in the first round of the amplification of PCR of 15 to 30 cycles. The products of the first round of amplification are then subjected to a second round of amplification using the second set of primers. The second set of primers anneal to a sequence internal to the sequence amplified by the first primer set. The increased sensitivity arises from the high total cycle number, and the increased specificity arises from the annealing of the second primer set to sequences produced by the first round. And then we have digital PCR. Normally, PCR exponentially amplifies nucleic acids, and the amount of amplification cycles and the amount of amplicon allows the quantification of the starting quantity of targeted nucleic acid. However, many factors complicate this calculation, often creating uncertainties and inaccuracies, particularly when the starting concentration is low. Digital PCR overcomes these difficulties by transforming the exponential data from the conventional PCR to digital signals that simply indicate whether or not amplification has occurred. Digital PCR is accomplished by capturing or isolating each individual nucleic acid molecule present in a sample within many chambers, zones, or regions that are able to localize and concentrate the amplification product to detectable levels. After PCR quantification, a count of the areas containing PCR product is a direct measure of the absolute quantity of nucleic acid in the sample. So this concludes our topic about polymerase chain reaction. Thank you and have a good day.